You're listening to the Common Descent Podcast.
Hello, Will.
Hello, David.
Hello, everybody, and welcome to Common Descent, a podcast about paleontology, evolution, and the history of life on Earth. This is episode 232, the second to last episode of 2025. This episode, we are tackling the subject of vertebrate origins.
Yeah.
We will investigate the evolutionary beginnings of our big family tree. Vertebrate animals, animals with bones, kind of.
Yeah, yep.
We will explore who belongs in this group, who our close relatives are, and what we know from modern animals and fossils of how vertebrate organisms got started. It's going to be a lot of fun. We'll touch on a bunch of really famous fossil sites and fossils themselves in this discussion.
This is another one of those exciting topics of we talk about vertebrates all the time.
That's like what the podcast is about.
Yeah, exactly. Now let's actually talk about what we've been talking about.
Yes, this episode is also going to go very nicely alongside the Bones episode, episode 210, and the Teeth episode, episode 88. So that also a little bit of a spoiler for something we'll talk a little bit about. It'll also go very nicely alongside the Brains episode, episode 121. And
Excellent.
Tails, for that matter, 150. A lot of the things we've talked about are like vertebrate things, like
Yes,
stuff
that's
that is
very true.
vertebrate stuff. So
Yep.
we'll get into all that. But before we do that, we've got a whole first chunk of episode to do, starting with shouting out the people who requested this topic. Every topic we cover on the main episodes of the podcast is requested by our audience. This was a popular topic requested by Thomas, Theodora, Mr. Pockets, Lassie, Emma, Adriana, Stephen, Ari, Levin, Stephen, a different Stephen, Franz, Robert, and Gazeboist.
Thanks for the requests, everyone.
Thank you all very much. Please enjoy. Speaking of calling people's names out on the podcast, we have a Patreon. The support that we get from our patrons allows us to do everything we do with the podcast. Our Patreon is constantly growing and expanding. We're developing connections with our community through live streams and bonus content, sneak peeks at upcoming projects, director's notes, all sorts of cool stuff like that. And at certain levels on the Patreon, you get your name shouted out here on the podcast. This episode, we would like to welcome to the Patreon Joseph and Percy.
Welcome. Thank you for joining our Patreon.
Thank you to everyone who supports us on Patreon. If you, dear listener, are able and willing, please consider joining us on Patreon. You get all sorts of cool goodies, and it helps us keep the lights on, as it were.
Yes, quite literally.
The lights in this case mostly being like Podbean and like gas in the car, all that kind of thing. The lights in the car. It is almost the end of 2025, which means that it is almost time for the end of the year Q&A. As of this when we are recording this, the Q&A form is closed, and we will soon be sitting down to record our question answering marathon that we like to do at the end of every year. That'll be coming out right at the end of the year, so keep your ears out for that and be prepared to listen to us talk for 5 hours or something. Thank you to everybody who submitted questions for that. Also coming up, the following month, the end of January, is going to be the 9th anniversary of Common Descent.
Yep.
It'll be fun. We'll have a live stream like we often do. We'll make some announcements about what's coming up later in 2026. And maybe we'll have some other special stuff that we'll do celebrating. We're planning on taking it a little bit easy for this anniversary because the one after this is going to be like a big deal.
Yes.
But we will have a live stream. We will have announcements. We will be doing some special stuff. So stay tuned for that.
Always a ton of fun.
And we got mail.
Yeah, we did. We got a postcard from Zelfia, who came back from a trip to Morocco, where they were excited to be able to recognize some fossils at a seller had there, and sent us a couple of polished fossils, two pendants, like they have holes in them to be able to be put on like necklaces or whatever we want to. And one is a round one with an ammonite, and It looks like one ammonite flat on, and then one like that's an edge of an ammonite
Neat.
is there as well. And then the other is an orthocone, the cone-shelled cephalopods. And they're very cool, and so cool that Zelfie was able to like use podcast knowledge on their vacation.
Yeah, thank you so much for thinking of us and sharing your gifts are great. Also, just like a long postcard message is delightful.
It's the best. It's just wonderful.
With all that said, let's get on to the news. Before we get into our main topic, just like every episode, we're going to cover some recent news in the world of paleontology and related sciences to see what's happening on the cutting edge. Will, what is new?
My first news is an update to last episode, which is news about how to detect whether fossil animals were aquatic.
Oh, hey, this would have been great
Yeah.
for 2 weeks ago.
this gives support to some of the questions we brought up in that episode.
Incredible.
This is research by Caleb Gordon et al. In Current Biology, and the press release is by Jim Shelton from the Yale University and phys.org. So, as we discussed at length in last episode, many different lineages of animals have returned to the water and become secondarily aquatic tetrapods, amniotes. But while it is usually quite easy to tell a fully marine adapted animal because they're shaped like a streamlined, dolphinish, sharkish shaped animal, the semi-aquatic and more aquatic, how aquatic the more ancestral and earlier members of those lineages are, can often be quite difficult because there are some animals out there that at a glance would not for sure look like a swimming animal and yet could have been.
Mm-hmm.
There's been lots of different ways. We went over a number of the different physical attributes that can give hints as to the aquatic nature of an animal. This study is trying a new way to approach that using phylogenetic machine learning tools.
Okay.
So basically, creating a learning program that will be able to look for patterns and then apply those patterns. And this allowed them to test out which pattern was the most accurate to go with. They wanted to test previously suggested bone indications for, marine adapted limbs or aquatic adapted limbs, whether or not there was webbing on the digits of the hands and feet, whether there was soft tissue flippers and stuff like that. They collected more than 11,000 original measurements from amniote limbs, so amniotes being reptiles and mammals, and used these to train the phylogenetic learning models. And then they were able to go through and select which one seems to be the best predictor models. And they found that hand length seems to be the best prediction.
Okay, that makes sense.
Makes sense.
I'm thinking of a whale's hand.
Yeah, exactly. They said that using this, they were able to predict soft tissue flipper, like the presence of soft tissue flipper structures and aquatic habits with more than 90% accuracy among the amniotes they looked at. So this is
Cool.
looking at modern groups that we know what their behavior and what their soft tissue is, and now using this model to predict fossil groups. They placed this onto the phylogenetic tree of amniotes and saw how some fossil members fell out, and they found some interesting trends, some which make sense, some which contradict some long held or previous estimates. Some of the ones that made sense, they found highly aquatic habits for all known ichthyosauromorphs. So the group that includes ichthyosaurs, they all seem to be aquatic quite heavily. That makes sense. They found evidence for multiple independent origins for aquatic habits among sauropterygians, which is the group that leads to plesiosaurs, mosasaurs, the aquatic lizards, and theropod dinosaurs, which we'll talk about more in just a second.
Huh.
Which would also include birds. So,
Sure. Yeah, penguins and
Yes.
water birds and stuff like that.
But there is some interesting notes there. One of the big ones that's like, I won't be surprised if there are responses to, they did find that the mesosaurs, the famous first aquatic tetrapods were semi-terrestrial,
Interesting.
that their limb proportions do not support an aquatic lifestyle, that they were probably still swimming, but more like an alligator, still
Sure.
coming out of the water quite often, spending decent amounts of time on land, still very heavily tied to the land. They were quoted saying they didn't completely leave the land, they were not aquatic.
Mm-hmm.
They also found that that's true for all Paleozoic reptiles that had been suggested to maybe be aquatic. That it doesn't seem like anyone before the Mesozoic, at least in reptiles, could be considered secondarily aquatic. And then the other one that is also going to get, I'm sure, quite a bit of attention is they did have a conclusion for Spinosaurus based on this model.
Sure.
As we talked about in the last episode, and as is famous, the evidence for how aquatic Spinosaurus is has disagreed with each other depending on what study and the way they're interpreting the aquatic lifestyle. Some suggesting that it was more like a wading bird, like a crane, spending time in the water, but like at the shore, while others have suggested that they should be able to swim and dive and maybe even pursue prey. Based off of this model, it suggests that they spent, and this is the quote from the researcher, a vast majority of its time submerged.
Interesting.
That it was a swimmer.
Mm-hmm.
So they called it highly aquatic habits, which is yet another brick onto that very interesting wall.
It's a very interesting. This is your comment is very apt. I think that this will for sure have responses and disagreements and other research that comes out to build on it. Not only because it disagrees with some previous interpretations, but also because it is interpreting largely based off a limited part of anatomy.
Yes.
Mm-hmm. Right. They said that the most useful thing was the shape of the hands. And one of my first thoughts is, well, you're not going to be able to identify a sea snake.
Yes, exactly. Yep.
And with theropod dinosaurs especially, I'd be curious to know, how does this trend in the hands associate with or maybe clash with the fact that a lot of theropods had reduced arms?
Yes, exactly.
If you're in a lineage that already has reduced arms or already has longer arms for some reason, that's an interesting set of results.
Mm-hmm.
I'll be curious to see if this same sort of model can look at other attributes.
Yes.
I'd be fascinated to see if they looked at like bone density or the detailed shape and proportions of other bones.
Yeah. And they mentioned about like this kind of approach could be used and tested to see its robustness against other similar transitions like bipedalism in our ancestors and the ability to fly in bird ancestors of how many theropods did get close to or maybe achieve flight. They also noted that this used one interesting aspect about the way they used to detect these features was a technique developed for World War II to interpret radar signals and whether or not an incoming signal was an enemy plane, using that calculation data for interpreting the limbs of these potentially aquatic animals.
That's pretty neat.
Right? So I'm very excited to see where this kind of approach might go or get refined, because if this is solid, this seems like a really potentially awesome tool for us to use in other anatomical interpretations.
Yeah. Well, then you're going to love my first news, because
Excellent.
my first news is also using machine learning programs to identify attributes of fossil remains.
Yes. See, this is the kind of teaching the machines that I'm all about.
This is like good artificial intelligence.
Yes,
we're doing
absolutely.
it. We're absolutely, this is great. In this case, specifically trying to identify some of the earliest signs of life on Earth.
Ooh.
This is research published by Michael Wong et al. In the journal PNAS, and in the blog post, we will link to an article by Michelle Starr on Science Alert. As we have discussed before, signs of life on Earth go back over 3 billion years, but it gets tricky. There are ancient fossils like stromatolites, but a lot of the earliest potential evidence for life on Earth is chemical evidence. It's molecules that seem like they were formed by some sort of living thing. These can be really tricky to identify, partially because they are literally just molecules, and it can be very difficult to tell a molecule that was formed by a living thing versus not by a living thing. It also is difficult because these break down over time. These traces also become fossilized, and that fossilization process can alter the shape and structure of them. So, in this study, a team of scientists, which include astrobiologists as well as geologists and others, started by collecting 406 samples of biomolecules from well understood fossils, modern examples, and synthetic examples, and then analyze them using gas chromatography and mass spectrometry, which basically means you take these molecules and you vaporize them and then measure the components in that vapor. There's more to it than that. Chemists can come at me. Then, with all of that data, they trained a machine learning algorithm to identify the differences between different types and formations of these molecules. Basically, the goal here, like the study that you just described, is there should be differences between these samples because they were formed in different ways. Are there differences that we can't identify? But if you feed a bajillion variables into a computer program, it can spot those differences.
Yes.
So they train the algorithm to identify differences between biotic and abiotic samples, so formed by living processes and formed by non-living processes, plant versus animal origins, and photosynthetic versus non-photosynthetic origins. Once the models had been trained, they tested them on known samples, and they reported that the model was able to accurately identify these categories between 95 and 100% of the time.
Awesome.
Which seems pretty good. Then they turned this algorithm onto some fossil samples that are less certain. They found that in samples that were less than 500 million years old, so like Phanerozoic eon, they had a clear biotic signature. Like, no question, these fossils were biologic in origin. That makes sense. That's what we'd expect. Older than that, it gets a little bit less clear, largely because A, we don't know the identity of all of these samples, but again, the samples degrade and change over time. Among their samples, the oldest sample that came back as positive for biogenic in origin, right, biological origin, came from the Josephsdal Chert in South Africa, which is around 3.3 billion years old, which makes it nearly as old as the oldest stromatolites, the oldest fossils.
Cool.
They also found their oldest evidence for photosynthesis in these biomolecules were samples from South Africa and Canada at 2.5 and 2.3 billion years old, which, if accurate, breaks the record for confirmed photosynthesis in the fossil record by almost a billion years.
Whoa. Oh, that's awesome.
Now, they did test older samples. They tested, I think, as far back as 3.7 or 3.8 billion years, and their program did not identify any as being biological in origin. But they do note that that could be because of breakdown of those molecules over time, things that are happening in the fossilization process. So that doesn't necessarily mean there isn't biologically, or those samples aren't biological in origin, but that this program in its current state can't identify that.
Yeah.
From this, just like the last one, individual study, this seems like a very promising start for helping us to differentiate between where certain molecules came from, which is especially valuable when we're trying to study early Earth, the evolution of photosynthesis, or I wonder if this could do like eukaryotic life versus prokaryotic
Ooh.
life, stuff like that, that I didn't see mentioned in this study. Very useful for studying the origins of life on Earth, but also elsewhere.
Mhm.
If we can turn this program onto samples from Mars, we might have similar results. That might be able to help us detect if there are molecules of biological origin elsewhere in the solar system.
That's so cool. I think one of the things that I find most fascinating about tools like this, these machine learning models and analyses, is it can take a look at a body of data that we can't comprehend and process. And then when it finds a pattern, we can ask, wait, what pattern? Like, how were you able to distinguish? And now we have a new lead that we
Mm-hmm.
can hopefully then start problem solving with. And it's very exciting. It's also, I like admire these studies for taking on the workload of feeding in the just absolutely mind-boggling amount of data that they have to get these things to potentially start spitting out answers, and that's very cool.
Yeah, this is really, we talk a lot, we get questions a lot from people who listen to the podcast who like to ask about how our field of science has changed over time,
Mhm.
because so much of paleontology is very old school. It's
Yes.
literally pulling stuff out of the ground and then measuring it. These kinds of programs and algorithms are really, really a new thing. This is very 21st century, that we're able to say, all right, here's a bunch of data, run calculations that would take a human being hundreds of years to do.
Yes.
Right? Do that in an hour and then tell us what you found. That's really helpful. That it's and especially when what we have to work with is such limited
Mhm.
information, and so much of what we're doing is comparing between different physical samples. Having something that can go in and go, yeah, we've analyzed this from every statistical angle you could, here's what the patterns are, that's a huge help.
It's awesome. So cool. Let's go find alien life.
Bring this robot to Mars, install this algorithm on
Yes.
the next rover. I'm sure that's not how it works, but
Yeah,
that's
yes.
for my version of this. That's what I want you to do. I want you to teach the next Mars rover to do this.
Put the AI in the atomic rover.
That's what could go wrong.
Well, my next bit of news is a study revisiting a famous fossil, Dunkleosteus, to look at the skull and jaw musculature and trying to figure out, trying to get a better idea of how its jaws actually functioned. This research is by Russell Engelman et al. In the Anatomical Record, and the article is another press release from phys.org from Case Western Reserve University. So Dunkleosteus is famous placoderm fish from the Devonian. It's like 360 million years old. These were roughly great white sized animals, like 14 feet longish, famous for having that armored face and instead of teeth, sheer like sharpened armor mouth parts. The questions of how this very impressive and very unique mouth functioned have been, have surrounded Dunkleosteus the whole time. And there's quite a bit we don't fully understand about its skull anatomy and jaw structure. In the article, in the press release, the authors note that the last major like examination of the bones and everything was in like the 1930s. That
Mm.
there've been models since then, but not looking directly at the bone, modeling off of what we already knew. They were wanting to return to the bones and apply new specimens that have been discovered since then to try to give us a better, more detailed look at what the structure was actually like. Part of the reason this has been difficult is that placoderms were cartilaginous fish. Like sharks, their bodies are not made of a bony skeleton, it is a cartilage skeleton. So the bony plates on the outside of the body preserve, but a lot of the structure inside does not. Since then, though, we have found some specimens where the cartilage has preserved. And over the last, century or so, we have just found enough of those for them to piece together a more complete idea of what was on inside Dunkleosteus's head. They found some interesting notes. First, that based off of those rare specimens and preserved attachment sites on the bony elements of the skull, as well as comparing it to other arthrodire placoderms, which is the group Dunkleosteus is from, they found that its skull contained way more cartilage than previously was suspected. Nearly half of its skull is cartilage, meaning that major jaw muscle attachments and cranial articulation, joints of the skull, were located on cartilage elements. So muscle attaching to cartilage, not bone, and major flexions of the skull based off of cartilage joints. They were also able to get some more detailed information about what the muscle structure of the jaws was likely organized as. They noted one specific jaw muscle that would have run along a bony channel that is similar to ones we see in sharks and rays, and is now some of the best evidence for this feature in ancient fish.
Cool.
So just giving some information about exactly how the muscles work, but also how that feature has tracked in fish evolution. And this also allowed them to just get a more updated look at Dunkleosteus in general. And they noted that even though it's the famous placoderm, it's actually quite weird for a placoderm.
Right. That this is the thing that happens a lot.
Yes.
That we've talked about is that, like T-Rex. Like,
yes,
this is
exactly.
the famous theropod dinosaur, but it's unusual among theropod dinosaurs.
It is a standout member. They noted that Dunkleosteus has no teeth, has shear-like mouth parts, blade teeth, so to speak, as well as a lot of its cousins in the arthrodire group also have that feature. But most other arthrodires had teeth. So it's unusual in that regard. And that when they look at it in the broader evolutionary trends of the placoderm group, they note that this trend toward a blade-like mouth is likely adaptations for taking on larger prey, being able to take chunks out of bigger prey animals, and is a convergent evolutionary feature that we see in other arthrodire groups, and seemed like a evolutionary trend that was happening across many lineages of this group of placoderms, but separately.
Interesting. So Dunkleosteus wasn't original.
Yes.
It was one of a number of lineages doing this. It just became the big famous one.
Exactly. They also note that based off the skull anatomy, some prior hypotheses that maybe suction feeding was utilized don't seem to have as much support now with their interpretation.
So again, more like a great white shark, where
Yes.
it's you're going to the prey. You're not taking it to you.
Exactly. And on that note, they make the point, which is obvious, but I love this, that bite force estimates have been made for Dunkleosteus for a very long time because we want to know. It has a can opener for a face. We want to know how hard it bit.
How many cars could you crush?
Yeah, exactly. And they note that those will all have to be re-examined based off of this new, because we now have a different structure than those estimates were based off of. So those estimates might change not at all or drastically in who knows what direction. And that just overall, this really highlights the fact that very often the arthrodires, the famous placoderms like Dunkleosteus, are treated as kind of a very similar group, that the little ones were just little Dunkleosteuses, and that truly they were a very diverse and had many specialized members, and Dunkleosteus being one of those quite specialized and different members of the group.
Yeah. These kinds of studies are very fun because it can be very easy to get the impression that we discovered this thing 100 years ago, we studied it, and now we know it.
Yes.
And the reality is that we're still constantly returning to these and learning more about them and refining our understanding of them. And that, again, going back to the T-Rex example, these famous specimens and species that get a lot of focus and attention, that research, one of the most important outcomes of that research is how much we learn about other species.
Yes.
By comparison and by understanding those evolutionary relationships.
Well, and I also like these when they point out that, not like that we've been shirking our scientific duties in but that there isn't like a overseeing body monitoring what research is happening. So if something gets researched in, 1950s and then no one else has that as their scientific hyperfocus, then that might not get studied in that much detail again for decades, and other people who are studying things tangential to it, or parallel to it, but aren't, a placoderm specialist, might not realize, oh, I thought that was like solidly understood, so I was basing stuff off of it. I'm not a specialist in it, so I didn't realize that that's actually based off of very old research that probably could use some revisiting. And so you get these moments where someone goes, have we gone back and double checked that now that we have this new stuff, and go, we hadn't, and there's a bunch of new information that we just hadn't realized was at our fingertips.
Yeah. Get the machines on it.
Yes, robots.
Tell us everything about this. You get the robots in it. Well, my last bit of news is also about an analysis returning to a famous specimen and interpreting how its face worked.
Excellent. Where's synergy right here?
We're very consistent today. This is particularly looking at the noses of Neanderthals.
Cool.
This is research by Constantino Buzzi et al. In the journal PNAS, and we will link in the blog post to an article on Live Science by Christina Kilgrove. Neanderthals, our close extinct relatives, ancient humans that lived famously across Europe, and particularly in fairly northern parts of Europe during the Ice Age, where it got pretty cold. Lots of studies of Neanderthals have interpreted or made the assumption that they were adapted specifically for these cold, dry environments. But as time has gone on, a lot of these have been questioned or challenged, that they might not have been specifically cold adapted in the ways that we liked to interpret them. One of these common suggestions regards their noses.
Mm-hmm.
Neanderthals have been noticed to have pretty wide noses, and especially later populations seem to have very large nasal openings, that they were just taking in a lot of air through their nose. It has been hypothesized that they also might have had very large sinuses, and that all of this space in the nose and the nasal region might have been adapted for warming and humidifying the air coming into their noses.
Yes.
We do see similar adaptations in other species where their nasal cavity is working like an air conditioner or a heater or a humidifier, just getting a little bit of adjustment to the temperature and moisture that they aren't getting in their environment. But this has been really tricky to confirm for Neanderthals because there aren't any complete Neanderthal nasal cavities known. Especially all those little delicate bones inside the nasal region. Those are often broken or degraded or crumbled, and we don't get a clear picture of. This study performed an analysis on a famous Neanderthal skeleton from Italy. This is a specimen, it's known as the Altamura man, found in a cave in the 90s near Altamura in southern Italy. It's about 130 to 170,000 years old. It is a famous specimen because A, it is very complete. It is a fairly complete skeleton, and because it is covered in cave formations.
Oh.
The skull, there's a picture of the skull in the article, and it's just covered in cave popcorn. Like it's been assimilated into the cave.
Part of the crew, part of the cave.
And because of the way that it's been sort of overgrown like that, it has never been moved, mostly because researchers are afraid that it's going to be damaged in the move because it's covered in all these cave formations. That, of course, has limited what sort of research has been able to be done on this specimen. These researchers used an endoscopic probe inserted into the nasal cavity that took a whole bunch of pictures in there and allowed them to create a photogrammetric 3D model of the nasal cavity.
It's scientific. Up your nose with a rubber hose.
Yeah. And then, I mean, it makes me, and I don't know what the specifics are, but it does make me think of like going to the dentist or going
Yes.
to the hospital or whatever. And we have tools that are like, yeah, we're just going to shove this camera or this mirror
Mhm.
into your face orifices so that we can get a look at what's going on in there and we don't have to cut your head open.
Yeah.
This allowed them to look at the Neanderthal nasal cavity without having to cut the head open. As it turns out, the Altamira Neanderthal skeleton has a complete nasal cavity, including all these tiny little bones that allowed them to get us a first look at what did Neanderthal noses look like. One of the big focuses of this research was looking for these features that have been proposed that made Neanderthal nasal cavities different. Things like certain expanded portions of the wall of the nasal cavity, particular bony projections where they otherwise wouldn't be, or bony areas where that should be there but aren't there. Changes like that. And what they found is, generally speaking, these unique features don't exist.
Oh.
Where they had been predicted or modeled or interpreted in the past, this complete specimen does not actually seem to have those. And in fact, they note that overall, the Neanderthal nasal cavity is pretty darn similar to modern humans.
Mm-hmm.
There is actually very little different going on in there. So while Neanderthal faces and skulls are different from Homo sapiens, it doesn't seem to be respiration related. It doesn't see the differences don't seem to extend to the nasal cavity and the sinuses as far as we can tell. They seem to have been breathing basically the same way as modern humans.
Cool.
The authors in the Life Science article, the authors are quoted as pointing out that it's easy to, this idea of Neanderthals having these big noses has become a bit of a trope, right? It's become something that we expect from Neanderthals. But they point out that wide noses are actually pretty common among Homo species, the ancient species. And they also point out that wide noses like Neanderthals are also the norm among living humans.
Yep.
Like having slightly wider noses, wider nasal cavities. Really, the one major area that has narrower noses are European populations.
Yes.
And that is something which is interesting, both in the sense that it does feel a little bit like, well, maybe our scientific bias got in there. And we were like, well, Neanderthals have different noses than us. And then most of humanity went, well, different noses than you.
Yes. Yeah.
Your nose is an unusual shape. But it's also interesting because it suggests that European Neanderthals and European Homo sapiens did develop different facial structures.
Yeah.
That maybe European Homo sapiens and European Neanderthals were maybe breathing a little differently or had some sort of different adaptation for surviving in that same environment.
Well, and that's something that because I remember the first time, like as a kid, I heard about this and I was like, oh, and then I would hear it over and over. And the thing that always kind of caught for me and didn't line up was I am always hesitant when looking at Homo species, especially, but for sure Neanderthals and our ancestors of like those are just people. And while like deep ancestrally, absolutely it makes sense that we would have physical adaptations for where we live, but once we get to the tool using levels that we were at that point, there's always a part of me that goes, okay, but couldn't just a cloth?
Right. But did you just wear clothes?
Yeah, like couldn't just a face mask have done the same thing? Like surely we could invent a way around the need for that seemingly minor of a physical adaptation. Like this is a very slight, it falls within the range of our modern diversity of Homo. So like surely there are things that would be more useful that we could make that would achieve that. So I'm always hesitant when it's like, if we look, you'll see this physical thing that we think is an adaptation to this lifestyle. And I go, maybe, but I think putting that as the final answer is taking a simplistic view of that group of people. Like, those are
Yeah.
people. And we have people nowadays that are living in parts of the world that their ancestors were not from originally and doing just fine. So I always hesitate on that.
It's a tricky tightrope to walk interpreting anatomical differences between different cultures and different human species and different human lineages, which are there. There is anatomical
Absolutely.
variation, but then being very careful about what we interpret that means about the people that
Yes.
live in those cultures or live in those populations. This is something that science has run into many issues with in the past among modern humans, is looking at these slight anatomical differences and going, this means that these people are fundamentally different from these other people.
Yes.
And that is not the case.
Yeah, exactly. And when it comes to humans, to peoples, we have, our tool use has removed us from so much of the typical natural selection system. And so, like, we break the rules in a lot of ways, which means that, like, features like that, could that just be what Neanderthals thought was handsome? And
Mm,
so, like, was that just what was appealing to them? So that's why it was a common feature, because it's like, well, yeah, that, I think that's hot.
right.
And so, of course, I'm going to hook up with people who have these features because that's what I like. And, like, that has come up with human cultures of, like, well, why is this culture that's what they fancied?
Yep.
Historically, that culture fancied this food, clothes, whatever it was, and it's different than yours because it's different peoples, different history. Doesn't necessarily say anything genetically or biologically about them, just different culture.
Yeah. Well, with the news out of the way, let's move way deeper into our evolutionary history than our fellow humans.
Past the nose.
Past the nose. We're going in deep. You get that extension on that endoscope. We're going all the way.
I can picture this transition in a documentary of the endoscope just continuing and wormholing.
The image in my head is an eye with the finger. We talked about this. This was a couple years ago. That eye with its finger just all the way down into the throat. What did I eat? Something's caught in my throat, and I'm gonna pick it out through my nose. Anyway, thank you, listeners, for joining us. We hope that you are having a great time with our nonsense. After a short musical interlude, we're going to go back to the origins of vertebrates and talk about how our lineage, how fish basically got started. Stay tuned. Vertebrates are the animals that we talk about on this podcast more than any others. And when we talk about vertebrates, we typically very quickly will define it, breeze by, and say animals with bones, which mostly works. That is a mostly perfectly functional definition of vertebrate animals. But for an episode where we go into the origin of vertebrates, we're going to have to get a little more detailed and specific than that. Vertebrate animals include about 65,000 living species. That is ray finned fish, cartilaginous fish, amphibians, birds, reptiles, mammals. In a single word, vertebrates are fish.
Yes.
Fish and all of their descendants, including us recording this podcast and all of you out there listening to this podcast.
Us and fish like you.
This podcast is brought to you by fish like you. Vertebrates are in many respects that we talk about them a lot because we're biased, because we are vertebrates and that's what we're interested in. But vertebrates are also extraordinarily diverse. There are tons of us on the planet. In fact, vertebrates are the third most diverse group of animals in the world today, following only mollusks in second place and, surprising nobody, arthropods in first place.
Yeah.
Crustaceans and insects and all of them, they've got it going. Vertebrates tend to share a handful of specific features. Obviously, this includes things like vertebrae, the bones of the spinal column. Vertebrates tend to have a skull. These bones and skulls tend to be made of either bone or cartilage. Vertebrates tend to have a brain that is protected within that skull. Vertebrates often have gills, they have tails, they have complex digestive systems. A bunch of these features that we have in common with fish and all the rest are classic vertebrate features. There are, among living vertebrates, two major groups. There are the cyclostomes, which are our modern jawless fish, hagfish, and lampreys. And then there are gnathostomes, which are jawed fish, which is everybody else.
Yeah.
That's us. We are jawed fish, you can tell, because of our jaws.
Because we naff.
We naff with them. To position us alongside the other creatures that we share the world with, vertebrates belong to a larger group called chordates.
Yeah.
It's easy to forget that vertebrate and chordate mean different things because almost all living chordates are vertebrates.
Yes.
There are two other groups within chordates. There are the urochordates, aka tunicates, which are sort of sponge-like or jellyfish-like marine animals. There are about 3,000 living species. And there are cephalochordates, or lancelets, which are kind of flatworm or eel-like marine species. There are about 30 living species.
Wow.
These are our closest living relatives to vertebrates. And we'll go into a bit more detail on them in a little bit.
I was kind of bracing myself because you said like the 3,000 tunicate species, and I was like, all right, that makes sense. They're little, soft jelly like ocean creatures. Those tend to do well.
Yes.
And then when we were getting to the lancelets, I was like, all right, are you about to tell me there's like 5,000 of these that I've just never? And it's like, okay, no, okay.
No, there's like, you could hold every species of these in one hand.
Yes, because they are also very small.
They're also very small. Chordates belong to a bigger group called deuterostomes. And our fellow deuterostomes are hemichordates, which are a bunch of worm-like marine animals, which include acorn worms and some others, and echinoderms. Echinoderms is your sea stars, your sea urchins, your sea cucumbers, a lot of your sea things. Your 5-pointed, radial, symmetrical ocean critters.
Kind of crazy to think that we are more closely related to star-shaped critters than a lot of other things that have a face.
Yes. Well, and this is a really interesting thing. Looking at where vertebrates fit on the family tree, we are in this corner of animal life alongside echinoderms. And what happened is that echinoderms got weird.
Yes.
So we'll work our way back through these groups and look at these defining attributes. What actually puts us within these groups? Why are we in the corner of the tree that we are? One of the things that gets really interesting when you look at such deep-seated evolutionary relationships is that the resemblances become harder to identify.
Yes.
Comparing us to other mammals is super easy, right? It's fur, it's the structure of the teeth. Even comparing us to fish is pretty straightforward because we do have most of the same body parts as fish because our ancestry only goes back so far. But once you start comparing phyla of animal life, those similarities come from such distant and relatively simple ancestors that you have to really start digging deep into the foundations of anatomy to identify those shared features that mark relationships.
Yeah, it's like when you zoom all the way out to what separates animal and plant is the kind of cells they are built out of.
Yes.
Because that's as much as you can have in common when you're that zoomed out. we're starting to butt up against that kind of framework.
Yes. And when we're comparing animal phyla, the differentiating features tend to be the way they structure their body.
Yeah.
Deuterostomes, this group that is us and echinoderms and our closest relatives, are bilateral animals. That is to say, two ways symmetrical, right? You have a left side and a right side that are roughly equivalent. Already, I've lost the people who really like sea stars because echinoderms did break that mold. But ancestrally and developmentally, they are bilateral animals.
Yeah, like a lot of larvae of echinoderms are bilateral, and then they
Yes.
metamorphose into geometry.
Yes. Deuterostomes are bilateral, coelomic, which means they have an enclosed body cavity, like all of us have, and they have an anus and a mouth.
Yes.
That's crucial. I use mine every day. What separates deuterostomes from other bilateral body cavity animals with an anus and a mouth, like arthropods and things like that, is in the embryological development of the group, the anus forms before the mouth does.
Yeah.
That is how we differentiate our corner of the tree from things like arthropods and other major animal groups, like mollusks and such. Their mouth forms before the anus, our anus forms before the mouth. These are super fundamental aspects of anatomical development.
And it's one of those notes that probably could sound like we're splitting hairs but that is fundamentally like, are you building your puzzle from the edges or the center? Those are very different ways to go about building a digestive tract. So it is a fundamental difference.
Yes. And like we were just discussing, some groups, especially because those are such simple fundamental starting points, have changed them over time. Echinoderms being the famous example, they lost that bilateral symmetry in their adult stages. They become something radically different, which is important to keep in mind as we start talking about what defines chordates. Our major group, Chordata, has classically 5 major defining features. These are the attributes, and this is, you might remember biology class if you learned about how to identify chordates. These are the 5 distinguishing features that separate chordates from everybody else. They are the notochord, which is where the name comes from. The notochord is a stiff rod of protein that runs along the axis of the body. Basically, this is the support rod for how the body functions. This is where muscles attach. This is how chordates move, is they have this stiff central pillar down the body, and then muscles move against that stiff axis.
Yes.
Chordates also have a dorsal nerve cord, also known as a neural tube, which is a hollow tube of nerve cells, essentially, similar to our own nerve cells, that sits above the notochord, along the very back. Number 3, pharyngeal slits, which are narrow openings just behind the mouth that open into the outside world, connecting it to the body cavity of the animal. Number 4, an endostyle, which is a particular organ behind the mouth that produces mucus and in many helps collect like filter feeding particles and stuff like that. And finally, a tail.
Yeah.
We talked about this in episode 151. A true tail, which means a muscular extension of the body beyond the anus. Yes. These are the 5 fundamental defining features of chordates: a notochord, a dorsal neural tube, pharyngeal slits, the endostyle organ, and a muscular tail.
I feel like, because we've harped on what a shame it is that we lost our tails during evolution, and the fact that it's a defining feature of the group we're in, and yet we don't have it, I think this is one of the best times to drive home how much we messed up.
Well, I also think that this list, even further than that, really drives home how confusing it gets when you start looking at these fundamental defining features. Because
Yes.
not only do we not have a tail, we don't have any of those features that I just listed.
No, we don't.
In my body, there is not a notochord, there is not a hollow nerve tube, there are not pharyngeal slits, and there is not an endostyle, and I also do not have a tail.
That we promise you can check.
Because, like echinoderms, we have shifted quite a bit from where we started.
Yes.
Now, we will go into some detail about where those features went and how we are still chordates, even though we don't have any of those features. But before we do that, I want to go into a little more detail on our relatives, the other chordates, because that's going to set the stage really nicely for us. Cephalochordates, which are lancelets, which are also called amphioxi. You might have heard the term amphioxus. There are, like I said, about 30 living species. They are all at most a few centimeters long. They are tiny little guys. These are sort of fish-like. They have a long, narrow body. So if you think of a fish that's sort of flattened side to side, they have a long, narrow body with small fins along the margin of the body. They don't have like fins like pectoral fins or pelvic fins. They're not swimming with limbs. They swim by wiggling their body like an eel. Lancelets do not have gills. They do not have jaws. They do not have a distinct head. There isn't even in most fish, you can see where the head ends and the body begins. Lancelets don't really have a head in that regard. They also do not have bones.
Yeah.
They are fish-like, but they're missing a lot of the most important features that identify a fish. Lancelets are typically filter feeders that live on the seafloor. They're down in the sediment and stuff. And all 5 of those chordate features are pretty evident in these animals. They have a tail, they have the notochord, they have all of the bits.
Yeah, if you look up this list, you will see a picture of a lancelet with the arrows pointing to the body because it's the classic example.
Yes. Yeah, a lancelet, not only does a lancelet have all of the 5 defining chordate features, that's most of what a lancelet is.
Oh yeah, you described a lancelet when you listed out those 5 features.
Yes. It's like this is just the most streamlined, stripped down version of a chordate. It's got those 5, and that's about everything.
This is a level 1 beginner chordate. You haven't entered into the skill tree at all.
The other feature that lancelets have that is another important feature among chordates are blocks of muscle.
Yeah.
If you think of like a fish fillet, that like zigzag pattern in the tissue of the fish, those are muscle segments that are called myomeres. Basically, blocks of muscle that sit right next to each other that can flex against the central pillar of the body that move the body. Lancelets have that. That is another important feature because, again, like I said, that's what the notochord is for, in large part. It provides a stiff structure that the muscles can act against.
Yeah, if you don't have something like that, you're gonna move much more like a floppy animal like an octopus. And octopus can't like lift their arm out of the water because there's nothing for them to work against. That tough structure allows you to do things that you can't without.
Yes. Then we have urochordates, aka tunicates. These are, again, ocean animals. They tend to be sort of barrel-shaped and bag-like. They're gelatinous. They're kind of like a sponge or a jellyfish. They look sort of like a plastic bag floating in the water in a lot of cases.
That's exactly what I was thinking. Yes.
These are filter feeders as well. Some of them are sessile. They anchor to the seafloor like sponges. This includes things like sea squirts, which are tunicates. Others are floating and form colonies, like salps, which form these sort of stringy colonies of these bag-like creatures. Pyrosomes are also in this group, which are, if you've ever seen those videos of what looks like a floating bag worm that glows, those are pyrosomes.
Yeah.
And this also includes larvaceans, which are these animals that build these like mucus shells, kind of. They're called houses. that can be a meter wide, that they sort of float in the water column. These are larvaceans.
Cool.
The interesting thing to note about these tunicates is that most of them also don't have these chordate features. Like echinoderms, they lost them as adults. But all tunicates, all urochordates, start out as larvae that are roughly the shape of a lancelet.
Yes.
They look like a lancelet. They look like a tadpole, really.
Yeah.
Right? A simple little body, a strong tail with the notochord, the neural tube, the pharyngeal slits, the endostyle, the tail. They start as a classic chordate and then metamorphose into something that is very unchordate-like.
Into a bag.
Into a bag. The larvaceans actually do stay as vaguely tadpole-shaped.
Oh, cool.
Most of the other ones basically turn into a jellyfish, which is a very strange, which is also kind of what jellyfish do. The jellyfish also don't start as jellyfish.
Yep.
And then we get into vertebrates. True vertebrates. So lancelets keep all of that chordate structure their whole lives. Most tunicates are very classic chordate as larvae and then lose it when they metamorphose. Vertebrates lose their chordate features by the time they're born.
Yes.
In vertebrates, all of these chordate features are evident in the embryo. But in most vertebrates, most of those features have changed by the time they are even newborn animals.
Yeah. This is why you have that classic example of being able to show a bunch of embryos from a bunch of different animals and then look quite similar, because that's when we have those shared chordate features.
Yes. This isn't to say that our embryos look like ancestral chordates, because
No.
they also don't. They look like embryos. They're their own weird thing, but they do still have these fundamental features before they get changed.
Yes.
In vertebrates, we all start with that notochord, but as we develop, that stiff protein rod is replaced by the cartilage and or bones of the spinal column. Our vertebrae that give our group our name. The notochord develops, is replaced by those mineralized structures, and the remnants of the notochord develop into the discs between our vertebrae.
Which is so cool.
The neural tube develops into the spinal cord, which is enclosed by the spine itself, the bones and cartilage of the vertebrae. The front of the neural tube enlarges and forms an expanded structure that has multiple different sections, which we call the brain.
Yeah. It's one of those where we forget that a brain is a vertebrate feature.
Yes.
Lots of other things have brain-like neurosystems, but it's not a big lump like it is for us, because we did it in a special vertebrate way.
We put much of the bulk of our nervous system in one place.
Yeah.
Generally, the brain in vertebrates sits within a bony skull, because it's really important and we want to protect it.
Because we put the bulk of our nervous system in one place. And then someone went, wait, what about headshots? I went, oh no!
The pharyngeal slits, which are visible in our embryos, give rise to gills. And the gills in most vertebrates, which is to say fish, transition in early chordae, in like lancelets and stuff, the gills are used for feeding. They take in, or rather the pharyngeal slits, I should say, are used for feeding. In vertebrates, the gills that those slits have turned into are almost exclusively breathing structures. They've moved away from using them as feeding structures. Now they're for breathing. In many tetrapods, land-dwelling vertebrates, the gills have vanished and the structures that formed the gills have become parts of the jaws and throat.
Yeah.
We still have gill arches, and if you watch a human embryo develop, you can see those pharyngeal slits transition into these gill arches, and then those gill arches become part of our mouth and throat. The endostyle, that particular mucus-producing structure that's in our throats, seems to be the structure that gives rise to the thyroid gland.
Oh.
I think in lampreys, which are vertebrates, the larvae, if I remember correctly, the larvae have an endostyle, and then when they metamorphose into adults, it becomes the thyroid gland.
Oh.
The thyroid gland is a very important gland in our body that produces and controls a lot of hormone production. It's involved in running your body's metabolism and then growth and development. The thyroid's a big important deal.
Well, and it's cool because like it makes sense that it's become foundational to how we function because it's been there since our ancestry.
Yes.
It's like we've done a whole bunch with it.
And then a muscular tail, which most vertebrates just keep.
Yes.
That's just a feature that most vertebrates hang on to. A handful of weirdos have gotten rid of it.
Foolish fools.
Like Sicilians.
Picturing us like at a bar and then we look over and a Sicilian's there and it's like, hey.
Just I'd say, and then somewhere out in the world there's a Sicilian like reading the newspaper and he goes, hey. Vertebrates also tend to have a handful of other features. There are other things that have developed within the vertebrate group. These include, for example, paired sensory organs,
Mhm.
eyes, ears, nostrils, the lateral line system, these features that allow us to sense the environment around us, which tend to come in pairs and typically are on the head.
Yes.
Vertebrates tend to have fins, appendages, supported by muscles and mineralized structures. In fish, these are fins. In us, they're arms and legs. Same structure. And vertebrates tend to have more complex internal organ systems. The digestive system is split into multiple organs like intestines in the stomach. We have a heart with multiple chambers. We have a brain with multiple regions. This differentiation of organ systems is another thing that is iconic to vertebrates. As I mentioned before, there are two major groups of vertebrates that have gone different directions with them. The first are cyclostomes, which are our jawless fish. Lampreys, which, if you've never seen a lamprey before, they are kind of disturbing. Adult lampreys are long and eel-like. Their mouth is funnel-shaped because they don't have jaws. It's not a bitey mouth. It is a round funnel-shaped mouth full of teeth. Many adult lampreys are carnivorous.
Yes.
Either biting and feeding on things or sucking on fluids of their prey.
Just big old fish leeches.
The larvae of lampreys tend to be suspension feeders. So they're taking in particles and stuff from the water. Lamprey skeletons are mostly cartilaginous. They tend not to be bony. They have paired eyes, but they do not have paired fins. They're swimming with the body movement. And lampreys also have one nostril. So they have some of the iconic vertebrate features, but they do not have some other ones. The other group of cyclostomes are hagfish, which are very creepy.
Oh, yeah.
Hagfish are like slimy eels.
Yeah.
They are predators and scavengers. They have actually a skull that is cartilaginous. They have toothy mouths. They don't have, as far as I know, they don't have vertebrae. They also don't have paired fins. So again, lampreys and hagfish are both sort of a slimmed down concept of what a vertebrate is. The interesting thing about lampreys and hagfish is that they are also very specialized, which is to say, it is likely, and it is thought to be true by many scientists, that they have these simplified anatomy, possibly not because they're an early representation of vertebrates, but that they've lost some of those features.
Yeah, exactly.
That, like vertebrae, might be something that they started with and then over time lost. So there, it's hard to say how much a lamprey or a hagfish represents the ancestral condition for vertebrates because they have gone and done unusual things.
Yeah, they're living very unique lifestyles. Like many lamprey are dedicated parasitic predatory fish that by latching on, and then hagfish are like can tie themselves in knots and produce crazy amounts of mucus and slime. Really weird features.
Yeah. And then all the other vertebrates are gnathostomes or jawed fish. These eventually down the line develop true jaws with true teeth
Hmm.
made of dentine and enamel. Lampreys and hagfish have tooth-like structures, but they're not quite the same as the teeth that we think of in vertebrates. Jawed fish also tend to have a dermal skeleton, which can be armor and scales, but also includes a bony brain case and the vertebrae that protect the spinal cord, bony rays in their fins, and paired fins,
Hmm.
a
Hmm.
pair of front fins, and then a pair of back fins, which go on to give rise to the arms and legs of their descendant tetrapods. So, quick recap. Chordates are all vertebrates, along with lancelets, which are very chordate-like, and tunicates, which start out very chordate-like. And then vertebrates are our jawed fish, which is most of us, and our jawless fish, which today are lampreys and hagfish. There has been a lot of debate over the years about the relationships between the components of these different groups. For
Hmm.
example, whether or not hagfish and lampreys are related to each other,
Yeah.
which these days the evidence does seem to suggest that they are one group,
Hmm.
that they are closely related to each other, and then the rest of us are all in jawed fish. Similarly, there has been lots of debate over the years about whether lancelets or tunicates are closer to vertebrates.
Hmm. Hmm.
And most recent studies with genetics and proteins seem to suggest that tunicates are our closer relatives, which is fantastic because tunicates are the weird ones.
Yeah. Ha. That's really fun.
So, comparing the features of basal chordates to true vertebrates, one of the major themes that has been identified, one of the sort of major, when researchers are looking at it, they're going, this seems to be a major important thing going on here, is the development of the head.
Yes.
This is once again, this is a weird thing for us to think about because heads are such an iconic aspect of most of our animals that we're familiar with. But having a head is very much a thing that vertebrates did.
Yes.
It really drives home, frankly, how convergent vertebrates and arthropods are.
Absolutely. Yes.
That so many of the iconic features, like legs and heads and sensory structures, arthropods also did it.
Yep.
That
Yep.
is a discussion for a different episode. A paper back in 1983 proposed what has come to be called the new head hypothesis, that early vertebrate evolution was driven in large part by innovations in the head. That the
Yeah.
structure of a head was an important part of this evolutionary shift. The shift from the simple nervous systems like we see in tunicates and lancelets to the more regionalized system with a brain up front with different region, right? The forebrain, the midbrain, the hindbrain. Episode 121, we talked about brains. The concentration of sensory systems up in the front of the body, eyes and ears, as well as gills, the adaptations to the mouth. Vertebrates became very head forward in
Yes.
their anatomy. This is possibly associated with a shift in feeding style. A lot of our living chordates that are not vertebrates are filter feeders.
Yes.
And it's thought that one of the things that might have led this shift in vertebrate evolution was a transition from filter feeding to predation.
Mm-hmm. Mm-hmm.
One of the things that a head is really great at doing is for sensing and grabbing and eating things.
Yes. Going and getting stuff.
There's also been a lot of research into the genetic basis of this feature. I'm not going to go into a whole lot of detail in genetics and development because I don't want to. But this is an important piece. This expansion of cranial attributes seems to be related to a developmental feature called the neural crest.
Mm.
The neural crest is a special group of cells that develops with invertebrate embryos. This is a group of stem cells. And early in vertebrate embryo development, this neural crest of cells differentiate and migrate all around, giving rise to various parts of the skull, the nervous system, the cardiovascular system, sensory organs. This packet of stem cells in our embryos seems to be largely responsible for the development of heads as we have them in vertebrates. And there are simpler versions of a neural crest in lancelets and tunicates. It seems that this was a feature that became more prominently developed in vertebrates. There has also been a bunch of study of the genetic difference, right? The actual in the genome difference between our chordate relatives and vertebrates, which again, I'm not going to go into a ton of detail, but one of the big features that people have identified is that there is this major change in the arrangement of the genome and the size of the genome from other chordates to vertebrates. And genetic analysis has revealed there appear to have been multiple whole genome duplication events around the base of the vertebrate family tree. There seems to have been at least one whole genome duplication means the entire genome of the animal became copied at least once.
Yes.
Sometimes more than once. It's because you can get like a tetraploidy event where it got copied multiple times, but basically you just multiplied the full size of the genome, which can be very relevant evolutionarily because it just gives you a whole lot more genes to work with.
Mm-hmm. Mm-hmm.
There seems to have been one of these duplication events around the origin of vertebrates, and then at least one more in each the gnathostome and cyclostome lineages.
Oh, separately.
Separately.
Cool.
This duplication of the genome is likely connected to the radiation of new features. This provided a lot more genetic scaffolding to work with and is probably related to the fact that vertebrates are so distinct from other chordates.
Very cool. That makes total sense. I was wondering if a genome duplication was going to come up just because of all of these special features. It's also really fun to get to note because, it's a thing that we just take for granted with most of the animals we talk about. Like, yeah, a head is just that's an animal thing. It's not actually. There's some specific groups that have done it, and it's happened convergently in different groups. And it makes sense that it is a big change because now you have a front and a back and a forward. Like now you can move very directionally. Like sea stars just move in whatever direction is the one that their senses tell them something is. They don't have a forward. They just move
right
in that direction. Now we have a face to work with. And well, it means now we have like a behind us, which is somewhat of an issue. We can also focus everything at that forward, and that allows you to do cool stuff.
I think it's really interesting to note that I mentioned at the top of the episode discussion the three most diverse living groups of animals: vertebrates, arthropods, and mollusks, all of whom tend to have a head.
Yes.
And the other category of animals that comes to mind when thinking about heads is worms.
Yep.
Having a head seems to be a winning strategy.
It is a really useful feature to be able to specialize. It can be useful to have, a generalized anatomy that can just handle things in a multiple ways, but specializing different parts for different jobs can allow those to get exceedingly good at those jobs.
Yes.
And that's very useful evolutionarily.
Like I said, thinking about what defines vertebrates and what makes vertebrates different from all other animals really does drive home our convergence with arthropods.
Yes.
That a mineralized structure that supports the entire body, paired limbs, paired sensory structures in a distinct head, we're kind of doing the same thing.
Yep.
But remember, they are mouth first, and we are anus first. And that makes us very different animal phyla.
I feel like it says a lot about the distinction between the two groups. Not gonna make any other distinct comments, but I feel like
Vertebrates are extremely concerned with getting rid of waste and excess.
and arthropods are here to bite.
They're gluttonous. All they want to do is take. We are givers. We return to the environment. Arthropods are takers.
And the ants say, yes, as you should. Give it to
Yes,
this is our planet. This is ours.
So, just by looking at the diversity of modern chordates and their relatives, by comparing genetic structures and developmental structures, just from that data, we get a pretty good sense of the evolution of vertebrates, of where we came from and how we got there. The steps, even though there are a bunch of unusual attributes, right? Lampreys and tunicates, and echinoderms, which have all done odd things and gone off in odd directions, as have we, vertebrates. The pieces are there to go, yeah, it does look like the larva of other deuterostomes, lancelets and tunicate larvae, early jawless fish, and then our embryologic development gives us a pretty darn good picture of the steps that it took to go from non-vertebrate ancestors to the earliest vertebrates. And that's before we even get into fossils. It's one of the coolest things about, this is true across evolutionary studies generally, but vertebrate origins is one that I think drives it home really nicely. If we had no fossil record, we would still have a really good idea of how vertebrates evolved.
Yes, we would still have a pretty reasonable estimate of what our ancestors, features they had, and maybe even some of what they looked like and were doing, because we've got so much of the roadmap of the evolution that has happened just in the extant members.
Yes, which is convenient because the fossil record of early vertebrate evolution is extremely confusing and frustrating. We'll get into that after a short musical break. Gnathostomes, jawed fish, and all of their descendants, including yours truly, have, generally speaking, an excellent fossil record.
Yeah.
Vertebrates, tetrapods, we have so many great fossils of vertebrate evolution. Other chordates and other vertebrates, not so much. I'm just going to run through some of our relatives, because what we would like to be able to do is look at our relatives in the fossil record and see what that helps us understand about early evolution. Unfortunately, lampreys, there are fossil lampreys. There are a handful of species known in the Devonian period and the Carboniferous period, and later than that. Hagfish, there are some potential hagfish fossils from the Carboniferous. There are definitive fossils as early as the late Cretaceous.
Okay.
So there's not a lot to go on, and it only goes back for cyclostomes to around the middle of the Paleozoic, which does tell us that hagfish and lampreys were established by, 400 million years ago or so, but that does not help us very much with the actual early evolution of the vertebrate lineage.
Yeah, but we were kind of vertebrate-y by that time.
They already made it.
So, not a lot to go on.
Tunicates, as you might expect, being bag creatures, don't have a great fossil record. Some modern species actually have tiny little spines that they build the body, kind of like sponges have.
Yes.
These little spicules that are mineralized structures. Those are known from the fossil record all the way back to the Triassic period. Those are actually, I don't know how common they are, but they are not unheard of. We have a number of examples of that. Before that, we basically have nothing. There are a few possible tunicate fossils from the Cambrian period. And having things from the Cambrian period is exciting because that is around the time we expect that chordates and vertebrates were really showing up for the first time. There is a fossil called Chungkongella from the early Cambrian of China, which was identified decades ago as maybe being a tunicate, but has since been disputed. More recently, there is another fossil called Shankoclava, also from the early Cambrian of China, which was identified back in, I think, 2003. Very similar to modern sea squirts, but more recent research has pointed out that there are some features that it has that don't necessarily line up with tunicates, so it might be one. The most recent candidate, identified in 2023, is a fossil called Megasiphon from late Cambrian deposits in Utah. This is a preposterous fossil. Megasiphon looks like you pressed a sea squirt into a book. It's preposterously good.
Wow.
It is extremely similar to modern sea squirts. It appears to be the oldest definitive tunicate. From my perspective, being a person who knows so little about tunicates that I do have to Google them every time I think about them to remind myself what they are.
Yep.
Looks like a tunicate to me. I'm convinced. But of course, give it a decade and maybe, there'll be more disputes about it. If these are tunicates, if one or all of these are tunicates, what it suggests is that the basic tunicate body plan and even life cycle seems to have been established within the Cambrian.
Yeah.
By the late Cambrian at the latest, we had tunicates living and functioning more or less like they do today, which again isn't super helpful.
Yeah, it's got kind of it's got kind of a double-edged sword here of like, I'm not surprised there are fossils that we're struggling to say whether or not they're tunicates, because when we were describing tunicates, we were like, kind of like a sponge, kind of like a jelly, kind of like all these other ocean creatures, because being a floating filter feeder is kind of the thing to do in the ocean.
Yes.
Like that's just the way most of your small organisms are surviving in the ocean, because that's what there is to do out there in the blue. But then if they are tunicates, then it's like it's got the bat issue of, well, you're already a bat, so
Yes.
I don't know how you got weird for a chordate, so we're still stuck.
It is nice because it does give us a calibration point. It says if this is a true tunicate, then true tunicates evolved by this time. So It helps us to put the timing of things, but it doesn't give us a lot of insights into the features and structures of the very earliest examples. And then, to round off our chordate discussion, there's lancelets. As far as I'm aware, there is one possible example of a lancelet from the Cambrian, and I think that's the entire lancelet fossil record. I think we don't have any lancelets. I could be wrong about that, but as far as I looked, I found one example of what might be a lancelet.
I mean, they're not robust critters.
No. And this really is when we look at all of these groups, with tunicates, lancelets, and cyclostomes, all of them are mostly soft tissue, swimming or floating, and very small, which are all terrible ingredients for making a fossil record.
Yes.
You're small, so you're going to fall apart or be easily missed. You're soft, so your body's going to decompose very quickly. And you swim, which means you're not in the sediment.
Oh, yeah. Like, you might not even make it to the seafloor because you just get picked apart by things on the way down. So it's just, you're really trying to not be remembered.
Yes. This does not bode well because we would expect the earliest chordates and vertebrates to be very lancelet or lamprey-like. However, there are several fossils that seem to represent early members of the chordate and vertebrate lineages. Most of these, in fact, almost all of the research that has been done on the earliest fossil examples of chordates and vertebrates has been done on 2 fossil sites. One of them is the Burgess Shale.
Yep.
Episode 89, the Burgess Shale is a Middle Cambrian site in British Columbia in Canada. It's about 508 million years old. It is maybe the world's most famous lager stud. It is a site of exceptional preservation. It preserves tiny, soft-bodied organisms in these deep ocean waters. The conditions of the Burgess Shale seem to have been very good for preserving soft things that don't typically fossilize. The other site is the Chengjiang fossil site in southern China, in the Yunnan province, which is early Cambrian and very similar to the Burgess Shale. This is another site of exceptional preservation. Chengjiang is a little older. It's about 520 million years old. Both of these sites have been extensively researched because they are our two best looks at ecosystem structure in the Cambrian, particularly when we're looking at the origin of most modern animal groups during the Cambrian explosion. These are the sites that we study. Most of the fossils there are, especially the soft tissue ones, are these carbon films. Just sort of, I mentioned the one tunicate that looks like it was pressed between the pages of a book. That's what these look. They are pressed into the sediment, and it's just a stain in the sediment in the shape and features of these animals.
Yeah, they look like charcoal sketches, like very detailed, fine charcoal sketches of the animals that are just the actual biological remains of the animal pressed between sediment layers.
Yes. Yeah, a charcoal is a good comparison because it is a carbon film. Although it does also make me think of investigator with a notebook being like, all right, tell me what the early chordate looked like.
This is their own chalk outline their body became.
That way we'll know where it was. The most famous example of an early member of the vertebrate or chordate lineage is almost certainly Pikaia. Pikaia was described back in 1911, one of the first famous fossils from the Burgess Shale, again, about 508 million years ago. Eventually, but these days we have more than 100 specimens of this fossil species, which is extremely nice. Pikaia was originally identified as a worm, as an annelid, but later reinterpreted, and an interpretation that has held up over time, as an early chordate.
Yes.
Pikaia overall looks a lot like a lancelet. It is a few centimeters long. It has this narrow, flattened body, right, side to side. It tapers at both ends. It appears to have been an eel-like swimmer, swimming back and forth. No fins, no defined head, like a lancelet, a small circular mouth, and it also has little tentacles on the head that seem to have been either sensory or feeding structures. It looks like a little lancelet-like creature, or like the larva of a tunicate.
I remember Pikaia confused the heck out of me when I first learned about them way back when, because they were like, yeah, lancelets are probably very much like what our ancestors were like. And as an example, here is a picture of what I was like, that's just a picture of a lancelet.
Right.
Like, and they're like, but now we're calling it Pikaia. And I was like, I believe you that these are different, but I don't understand how they're different. And I remember being very confused on like, am I missing up the names? Am I getting my names mixed up, or what's happening? Because they just look so similar.
And indeed, a lot of the debate about where Pikaia fits comes down to trying to identify those specific chordate features. Because lancelets aren't the only things in the ocean that are shaped like lancelets. Right? There are flatworms, there are mollusks, there's larvae of tons of stuff. So what researchers have tried to do is identify, does it have those chordate features? Fortunately, because this is the Burgess Shale, you get a lot of really exceptional soft tissue preservation, as this body was just squeezed into the sediment. Among Pikaia's soft tissues, researchers have identified what appears to be a notochord, that rod of protein down the body, what appears to be myomeres, those zigzaggy muscle blocks like we see in modern fish, what might be gills, the digestive tract, and what appears to be the neural tube along the back of the body, which, toward the front of the body, the neural tube appears to have a little bulb-shaped expansion toward the front. Again, a lot of these have been debated as to exactly what they are. Do they represent this? But they appear to be a smattering of identifying chordate features. However, it also seems to have some non-chordate features. For example, some researchers looked into it and found that the digestive tract that's preserved within the body seems to go all the way to the end of the body, which
Oh.
suggests that it does not have a tail.
Yeah.
It also appears to have been covered in a cuticle, this sort of outer coating of protein, which we see in lots of invertebrate groups,
Mhm,
but we
mhm.
don't tend to see in chordates.
Okay.
So Pacaya has this interesting mosaic of features that it seems to be overall very chordate-like, but not perfectly chordate-like. It has some unusual things that it either shouldn't have for a chordate or is missing for a chordate. There have
Interesting.
been tons of research put into this, tons of analyses trying to identify where does Pacaya fit on the family tree of life. There's been lots of disagreement, but overall, most research agrees that it is somewhere around the base of the chordate family tree. It might be an early chordate, it might be a cousin of early chordates, it might be the ancestor of it's somewhere around that root. Pacaya seems to be one of our best representatives of the beginnings of the chordate lineage.
It's always when we come across these like very early, very like Cambrian early of like this is the base of this major branch that like you can get these features that's like but it doesn't have this. It's like cool. Does that just mean it hadn't evolved yet, or that these chordate like features were common in closely related groups? And either one makes sense because we have chordates that are shaped like sea stars, so it's got this the range of things that can happen with that body shape can actually be quite vast.
Yes. Yeah, it's did that feature not evolve yet? Is this a lineage that lost that feature?
Yes.
Like lampreys and hagfish? Is this a lineage that convergently developed that feature? And this, as we've talked about before, when you get back to the beginnings of any group of life, the issue that you start to run into is that what separates the first members of a lineage from their close relatives is very, very little.
Oh yeah.
The very first chordates would have looked almost indistinguishable from their closest relatives that weren't chordates.
Well, and like you mentioned, had they did they lose features? We see in evolutionary lineages all the time where it's like this feature was ancestral, it was lost and then re-evolved later on.
Yes.
The current members and the ancient members look very similar, but that is not actually the same ancestral structure because it was lost and then came back in a new way. It evolution is not linear, and it is not clean, and it is not
Yes.
organized.
And this is part of the reason why, the classic way that we talk about evolutionary processes and the way that we sometimes teach about it is we talk about ancestors and we talk about the origin of a lineage. And what we want, because it feels satisfying, is for Pacaya to just be the ancestor of all chordates.
Yes.
But in the fossil record, we are basically never going to find the ancestor of a lineage. And in fact, there's no such thing as the ancestor of a lineage because even the
Mhm.
first members of chordates were a species, a
Yes.
population. So, what we really are looking for in the fossil record is a group of fossil organisms that represent the features that we're starting to develop. And somewhere within that group, true chordates arose, true vertebrates arose. We see this when we look at, for example, human evolution, right?
Yes.
Human evolution isn't a march from one species to the next. It's a group of species that are a little bit human like, and then a slightly younger group of species that are a little more human like, and then a slightly younger group of species that are a little more. That's how evolution works. It works in
Yes.
groups and lineages, not in individuals.
Well, because you can get cross-breeding with very closely related species and hybridization that can be viable. So we like this idea of, okay, but what species did we come from? Maybe we didn't. Like, this interbreeding mix of relatives could be what eventually gave rise to the population that eventually gave rise to et cetera, et cetera.
Yes. So what we would hope for is not just to have Pacaya representing our origin, but a group of Pacaya-like organisms
Mm-hmm.
from this time period. And fortunately, we do.
Woo!
There are a handful of other fossil organisms that seem to be living a similar life with a similar anatomy, possibly closely related. There is, for example, a species, the taxon Unanozoon, which is sometimes also Haikuella. I think Haikuella is a dismissed name at this point. This
Gotcha.
is a much discussed and much debated fossil from Chengjiang, right? The other Burgess Shale, the second site that we talk about a lot. Very similar to Pacaya, a few centimeters long, narrow in its body, appears to have a notochord, those muscle segments, appears to have paired sense organs.
Hmm.
It also has external gills, which is a little bit of an odd feature, and a segmented tail.
Interesting.
Unanozoon has this sort of odd mixture of features that is a different odd mixture of features than Pacaya. Nearly every trait of Unanozoon has been debated and
Oh,
disagreed
wow.
with. Even in like recent research, you'll see this species brought up in different regards. There's a lot of disagreement over how to interpret this animal, but like Pacaya, even among that disagreement, it is generally considered to be somewhere around the origins of chordates. Another group like this is a group of organisms called the Vetulicolians, which are known from both the Burgess Shale and the Chengjiang fossil site. These are vaguely tadpole shaped. The front half of the body is sort of big and roundish with a mouth and gills, and then the back half is this segmented tail like structure. So, quite tadpole like in shape.
Mm-hmm. Mm-hmm.
They tend to not have any apparent eyes or appendages. They tend to not have apparent muscle blocks, but some do appear to have something like a notochord.
Okay.
So again, somewhere around the base of chordates, these seem to be. Some researchers have suggested that these might even be on the tunicate branch.
Mm.
And then one more that I want to mention is a fossil called Cathemyrus, which is from Chengjiang, which has been considered possibly an early cephalochordate.
Okay. Okay.
It might be our only lancelet fossil. It is very lancelet shaped, appears to have those muscle segments, appears to have a notochord. So we have this collection of organisms from multiple parts of the world. We have them in North America, we have them in China, from the early Cambrian and the middle Cambrian, that seem to collectively represent this beginning of chordates and chordate relatives.
Cool.
Again, these are all tiny, they are soft bodied, they all appear to be active swimmers, all of which is going to make it harder to become fossilized. And the other thing that really is a limiting factor for understanding these early chordates is that they're only known from a handful of fossil sites,
Yes.
because the Burgess Shale and the Chengjiang fauna are two places that have the kind of exceptional preservation that generates fossils of these kinds of organisms, but those are very specific habitats.
Yes, indeed.
So we also have a limited vision of the ocean at the time, which really restricts how many of these organisms we're able to find. But there does seem to be signs of the early stages of chordate evolution.
Very cool. That is more than I was aware of. Like, more a wider diversity than I was aware of. That's very cool.
Yeah. And then we have a similar situation when we look at what appear to be the first vertebrates.
Yee.
Once again, there is a famous one. The famous one is Haikuichthys, which has famously been called the first fish. It's sort of the Archaeopteryx situation. Haikuichthys was described in 1999 from Chengjiang. It has since been expanded to hundreds of specimens. We have a pretty good record of this genus. Haikuichthys is a few centimeters long. Overall, in its shape, similar to cyclostomes, so like lamprey shaped. It has clear muscle segments. It has marginal fins, so like a dorsal fin and a tail fin and a ventral fin, so not like side fins, but fins around the outline of the body. It has gill arches. It has paired eyes. It has what might be nostrils.
Okay.
And appears to also have what seems to be a notochord, very important, cranial cartilage. So cartilage in the head, like hagfish, and fin rays. These mineralized, sturdy structures supporting the fins. All of these are things we see in modern jawless fish.
Yeah.
So Haikuichthys has been proposed to be a very early stage of vertebrate evolution.
Cool.
And like Bacaya, it's not alone. There are a handful of these. Also from Chengjiang, there is a genus called Mylocunmingia, which is known from a single specimen that has a head, gill pouches, muscle segments, a notochord, what looks like cartilaginous elements in the head and farther back in the body. So maybe an early take on skull and vertebrae. From the Burgess Shale, there's Metasprigina. There's over 50 specimens from the Burgess Shale and also at least one other nearby site. Paired eyes and nostrils, notochord, gills, muscle segments, but no fins and only a vague sense of a head.
Interesting.
And then I'll mention one more because this is fun. This was described last year in 2024, a fossil called Neanderthal from Utah, from a site called the Margam Formation, which is a lesser-known Cambrian Lagerstätte, a little bit younger than the Burgess Shale. Neanderthal is one fossil, a few centimeters long, roughly lancet-shaped, with muscle segments and paired eyes, but does not seem to have fins or a notochord.
Interesting.
Now, the authors do point out that the notochord rarely preserves.
Yes.
So it could be that it didn't have a notochord, but it could also just be that the notochord didn't preserve. One of the things that's interesting about Neanderthal is that it does have a head. It also appears to have a snout.
Oh.
The front part of the head is long.
Okay.
It has this like snout. What might be the oldest snout in the fossil record.
The oldest snoot that you could boop.
You could have booped this snoot back 500 million years ago.
That's so great. Well, and the thing that also keeps coming to mind to me as we're like describing these similar specimens that have sometimes some of the features, but maybe not all of the features. It made me think of I don't remember which episode this was on, but the news we talked about with turtle evolution, about how turtles seem to have evolved the different aspects of being a turtle, the top shell, bottom shell, and no teeth beak multiple times along different lineages. And it just turns out that our lineage did all three. But
yes, well, we get the same thing with birds.
Yes.
A lot of the features that define birds showed up in multiple lineages of ancient dinosaurs. Birds did all of them and then became the ones that we know.
Yes, exactly. So like that's the other thing that could be happening here is like, well, this one developed fins. This group did not develop fins. They were doing just fine with a flat body. This one developed a more snout, like these ones didn't, and just the ones we came from did all of those.
Yes. So again, we seem to have this global population of little lancelet-like organisms, some of whom seem like maybe early lineages of the chordate group, some of whom seem like early lineages of the vertebrate group, found from the early to the middle part of the Cambrian in a bunch of different fossil sites. It seems like in the Cambrian, quote, lancelet, was just sort of a regular way for animals to be. And that is where vertebrates got their start. Just piece by piece, these different features that define this group showing up and becoming more prevalent. Exactly what these animals were doing is difficult to know. Often their lifestyle is interpreted as being similar to modern plantslets or tunicate larvae, filter feeding and swimming, although again, with the development of the head, some of them might have been being more predaceous. By the time we get to the late part of the Cambrian and the early part of the Ordovician period, so we're in that 500 million, 480 million years ago time period, we start to see indisputable vertebrates, what we would look at and call fish.
Yes.
A lot of these belong to a group that is informally referred to as ostracoderms. This isn't one biological group. This is actually several different lineages. These are early armored fish. Many of these they have a distinct head and a long body and tail. The body and tail are segmented. And among this group of early jawless fish, we see a smattering of different features that are important. Some of them have paired front fins, pectoral fins. Some of them have large tail fins. Some of them appear to have gills that have transitioned away from any feeding function and are now structured like more familiar fish gills for respiration. Some of them appear to have a muscular throat that could have been used for suction feeding. And many of them have mineralized tissues.
Okay, yeah.
Ostracoderms are famous for having armored heads.
Yes.
The head is covered in these tough, often scale-like, sometimes tooth-like, bony armor plates. This development of true hard tissues, shifting from earlier cartilage to more tooth-like or bone-like structures, is a big deal for their ecology, a big deal for what they're able to do that provides structure for the body, it provides protection from predators, but it is even a bigger deal for studying early vertebrates because now we have a fossil record.
Finally.
Now these things are fossilizing like crazy. Ostracoderms are extremely prevalent from the Ordovician all the way to the Devonian period. For the early half of the Paleozoic, after the Cambrian, this seems to have been what fish were.
Which is
These
very
were
cool.
Still very simple. It was a head and a long body and tail. They have those segmented muscles. They have those fins. They have a bit of protection around the head. In many ways, these aren't that different from Haikuichthys and those earlier groups. But they do become very diverse. Ostracoderms include, for example, Galeaspids, which have these massive head shields. Pituriaspids, which have long snouts, this sort of long rostrum like some modern fish. Osteostracans, which have paired pectoral fins, front limbs. Phelodonts, which were not typically covered in bony shields, but instead with scales. And anaspids, who had no bony armor at all and were much more of just like a naked fish.
The ones who refuse to go with the trend.
Yes. Ostracoderms are very common and very diverse until the end of the Devonian period, the age of fishes, but by that time, some of these early jawless fishes had given rise to true gnathostomes. During the Devonian, we see the radiation of jawed fish. These include placoderms, which are another group of armored fish, as well as acanthodians, which are a group of cartilaginous fish. Among these groups, we see paired pectoral fins and paired pelvic fins. Now we have front legs and back legs.
Yes.
Mineralized vertebrae, mineralized fin rays, and jaws.
Yep.
True, proper, open and close jaws. These jaws developed from some of the gill arches from their ancestors, and then around that same time, we are witnessing the development of teeth.
Yeah.
For more on those mineralized tissues, check out episode 88, where we talked about the evolution of teeth, and episode 210, where we talked about the evolution of bone. These mineralized tissues start as armor and scales, and then gradually continue developing into things like those rays to support the fins, teeth in the mouth for grabbing food. By the end of the Devonian, vertebrates as we know them are poised to take over the world.
Yes.
We've got our ray-finned fish, we've got our cartilaginous fish, we even have our lobe-finned fish, who by the end of the Devonian have left the ocean. The evolution of vertebrates spans the Cambrian with Pikaia and Hycoichthys to the end of the Devonian, where we have the foundations for all of our major modern groups. And what's so fun about this is that it really does go through a smattering of lancet-like organisms in the early to mid-Cambrian, a smattering of lamprey-like organisms in the mid to late Cambrian, a smattering of jawless fish with different features through the first half of the Paleozoic, and then true jawed fish radiating in the Devonian. We do see this development of these features with a whole bunch of mosaics and experiments along the way.
Yeah, well, and it's fun because it is a very clear path as you watch the features slowly build up and the characteristics slowly show up, that you can see how we got from the beginning to where we are. It is a really nice evolutionary example of watching a major shift like that happen.
Yes. And it matches what we expect from modern studies, from
Yes.
developmental studies and DNA studies. Now, while I'm mentioning famous and important fossils in the study of vertebrate origins, there is one more group that I do feel like I have to mention because we just rarely ever talk about them on this podcast, and that's conodonts. Conodont is a name originally given to a type of fossil.
Yes.
These small tooth-like elements that are found all over the world throughout the Paleozoic, all the way back to the Cambrian and onward. These aren't just individual teeth, they're these apparatuses of multiple tooth-like structures. For many years, it was debated about what these were, whether they were true teeth like vertebrate teeth, what animal they might have belonged to. Eventually, I think this was in the 1990s, scientists finally discovered full body fossils of these animals. The reason we weren't finding those is because they were very soft tissued animals. Nowadays, there are a handful of these known from North America, Europe, and Africa. I think there's only like 3 or so, like full body conodont fossils.
Wow.
They appear to be jawless fish.
Yes.
Long eel like bodies, large eyes, rays in their fins, those supporting fin structures, muscle segments, notochords. They appear like the cyclostomes, like lampreys and hagfish, to be somewhere around the base of the vertebrate family tree. They might be cyclostomes, they might just be close relatives of vertebrates. And their teeth, those toothy structures, appear to be a convergent mineralized structure with other vertebrates. They're not true teeth. They seem to be a different thing.
Which, well, once again, the teeth inside a lamprey's mouth are tooth-like structure. Tooth-like structures are a very good thing to have in your orifice that you want to be grabbing stuff with.
Yes, and they're pretty easy to make.
Absolutely.
You just make like a sharp, tough thing.
Yes.
And now you
exactly.
have a tooth. Conodonts are known in the fossil record all the way to the Jurassic period. So they lasted quite a long time, and they seem to be part of this diversity of early experiments in vertebrates. So we have this pretty fantastic, clear trajectory. There are, of course, still a lot of open questions. The exact relationships between these different groups go back and forth. Again, these aren't relationship questions like, is it a fish or is it a squid? It's more, is it a true vertebrate? Is it just outside vertebrates? Is it just outside chordates? Exactly where does it fit? Are conodonts closely related to lampreys, or are they on their own branch? Those sort of details are still discussed and debated. The timing and origin of certain features are still things that we don't fully know, right? Exactly when did notochords become a thing? Exactly when did mineralized structures start to become common? Right, though the exact timing and rate of those developments are still things that researchers look into. And a thing that we know very little about is the lifestyles of these animals.
Mm-hmm.
How Pacaya and Haikuichthys and even conodonts were living. What were they doing with these new features? And could that help us understand why they developed the things that they did?
Yes.
There's a lot. If you look up recent research into this, like we'll put some links in the blog post. If you look at the most recent studies, there's a lot of discussion in there about detailed phylogenetic debates of where on the tree does this thing fit, when in the which features showed up first and which ones were established by this time and by that time, which are very informative details to sort out, but the overall picture is shockingly clear
Yeah.
of early vertebrate evolution, which is very satisfying because it's not always like that.
Absolutely. Well, and you making the point of like their lifestyles for many of the early vertebrates being a mystery makes a ton of sense because we talk about a bunch of when there's a fossil group that we don't have a modern example, like sauropods, long neck dinosaurs. We don't have anything really remotely shaped like that nowadays. So there's a bunch of mysteries, but you're still obviously a land animal. You're still like there's a bunch of things that are very intuitive.
Yes, you have a head, you have a
Yeah,
mouth, you have legs. Yeah, we generally know how that body works.
but like jawless fish, we've only got two examples, like general groups of what those are like today, and they're both weird
Very strange.
and not at all like what the more like armored. Nope, not even a little bit. And they don't have paired fins, so like there was a whole diversity of truly bizarre fish that did not have at least did not have a moving bottom jaw. Maybe they had like soft tissue that was doing crazy stuff inside their mouth, but when you see drawings they just look like swimming vacuum, like hand
Hmm.
vacuum cleaners.
Yeah, they're often interpreted as bottom feeders.
Yes,
They're just
exactly.
like slurping stuff up off the sea floor.
Which they could be doing lots of, they could be like puffing sand away and then slurping up. So they could still be hunters. They could be swimming through the water and suction feeding prey. So like there's definitely things they could be doing, but did you have a bunch of crazy soft tissue that was helping you do that? Did you have tongues that were coming into play? Like hagfish have a tongue that help them grab onto meat and get a hold of prey and stuff. So like what crazy structures did you have going on with these truly like alternate fish body shapes?
Yeah. These discussions are a lot of fun because it really does emphasize that armored jawless fish for tens of millions of years was what fish were.
Yeah.
That's what a fish, like if you were in the Silurian and it was like, let's go find a fish, that's what it meant. It meant to go find one of these bony vacuum cleaners. And before that, in the Cambrian, if you were going to go find a fish, it was a little lancet thing. A little
Hmm.
lancelet like Haikuichthys, a sort of eel-like or flatworm-like. That was this wasn't one individual or one species that was doing it. This seems to have been the diversity of pre-vertebrates or
Yeah.
pre-gnathostomes. And these days, fish means something different than it would have meant
Yeah.
500, 400 million years ago.
And we don't really have anyone that seems to be doing things the way they were doing things. So there's just so much to speculate on.
Yeah. For the rest of vertebrate evolutionary history, go listen to most other episodes of this podcast where we talk about all of that. If there's if there's a direct sequel to this, it's probably episode 77, which is
Mhm.
the evolution of tetrapods.
Yes. Uh-huh. Yep.
So go listen to that. And then eggs,
Yes.
episode 92, where we talk about the evolution of amniote eggs. So go enjoy, explore all of those things.
Yeah, we've never made a vertebrate playlist on YouTube and whatnot because it would just be most of the
Just
podcast.
all of them. I think there is room for a playlist that's like episode 100 and the origins of life.
Yes.
And then we haven't done like a eukaryote
Yeah.
evolution. So that's a little gap that we're missing. And then the Ediacaran fauna
Mm-hmm.
and the origin of animals. And then we've got this episode for the origin of chordates and vertebrates. And then we've got, bones and teeth, and then tetrapods. Listeners, feel free to put together what you think is the definitive life through time playlist
Well,
for the evolution of how things got to be as they are.
we could do a pretty decent playlist for from the origins of life to us.
Yeah, because we've got early synapsids.
Yep. Mm-hmm.
We've got milk.
Uh-huh. we could actually have a pretty decent playlist of how we came to be. That's cool.
Yeah, the episodes of wildly differing quality. Before we wrap everything up, there is one last thing to do, and that is our patron questions. One of the benefits that our patrons can receive on Patreon is the ability to submit questions for us to answer here on the podcast. Will, what patron questions do we have to answer today?
Our first question is from Jeff. With the preservation similarities between the mesolpit and the J-hole biota, each having anoxic water and volcanic activity, is it possible that we're seeing a very specific habitat that could possibly be not at all representative of the respective periods as a whole?
That is an excellent question and very relevant to this episode's discussion. We talked about the mesolpit in episode 160 and the J-hole biota in episode 152. Famous sites of exceptional preservation. Just like this episode, we talked about the Burgess Shale and the Chengjiang fossil site. The short answer to Jeff's question is absolutely.
Mm-hmm.
These are unusual habitats, and that's why they're able to preserve these incredible fossil specimens. And so, just like, and I saw this actually discussed a bunch in the papers I read for this episode preparation. The Burgess Shale and the Chengjiang fauna are great examples of Cambrian fossils, but they are probably not great examples of Cambrian habitats.
Yeah.
Same thing with the J-hole biota and the mesolpit. These are exceptional sites. And so we do, as paleontologists, have to be very careful about how much we're extrapolating.
Mm-hmm.
We can't expect that the whole world at that time looked like these exceptional fossil sites.
Yeah, well, it's there's a bunch of cool habitats you can look at that show you a really awesome diversity of like tide pools can show you a really amazing diversity of life.
Yeah, or rainforests.
Yes, but if you're like, so this is what the ocean is like, no,
Nope.
that's not going to tell you what the ocean's like. So this came up in the news recently, but I don't remember what it was, where they talked about the fact that we're missing some key environments that probably were the more important environments for that group they were looking at. And that's very likely the case with many of the organisms we're finding here, that this is probably not where they were doing best, because just odds are there are going to be other environments that we just don't have preserved.
Yeah, this came up in some of the papers about looking at, because the Burgess Shale and the Chengjiang fossil site are not the only Cambrian sites, and they're not even the only Cambrian Lagerstätten. And I've seen some papers with researchers who are pointing out we do have to stop relying just on these major sites. and
Yes.
do more research on other sites because we're going to get a better picture of global ecosystems.
Absolutely.
So the short answer to Jeff's question is yes, and the long answer is yes, and don't forget it.
Yep. Yeah. Because it's so, when you find a fossil site that is so well preserved, it's real hard not to want to just do all the research there.
Yeah.
Our next question is from Lydia, who says, "You have each been asked to write a Magic School Bus episode. What do these episodes look like?"
Great question. Very fun. And because we just finished this episode, I'm tempted that it would be the Magic School Bus Goes to the Cambrian period.
Yeah, that would be very cool.
To meet all of the cool, weird, early members of our modern groups.
That would be very cool. I feel like, now this is also because the last episode I prepared was secondarily aquatic,
Mm-hmm.
but I like when they do things about where they get to slowly move deeper and deeper into a topic.
Yeah.
And so, like, doing something like secondarily aquatic of, here's the first step to becoming a better swimmer, and then the next step, and then the next step, and they can keep encountering problems and going, oh, well, I can't do that. Looks like you need to lose your fur. And
Yeah.
it's that, or maybe like polar life or something, like one of those where there's a gradient that we can move along. I love those kinds of episodes.
I like the idea of doing an episode that the Magic School Bus becomes a fossil. And you go
Yeah.
through fossilization processes, and maybe different scenes or different characters become different types of fossils.
Mm-hmm.
That could be really fun.
Yeah.
Yeah. Really, I'm trying to think of things that the Magic School Bus didn't already do.
Right?
Because part of me is like, oh, explore like plate tectonics. But they did that, and it was awesome.
Yep. Yep. Um.
Or like the digestive tract.
Yeah,
you did that. They did the brain too, didn't they?
I think so.
Maybe the brain was... there was definitely a section for the brain in my Magic School Bus Inside the Human Body computer game.
That makes sense.
And
yeah, there's a lot of cool ones. Ooh, this would be a gross one, but I feel like if anyone could tackle it, the Magic School Bus could. Parasites.
Yeah.
And like emphasize, like, because that would be a fun one to be like, oh, but how many parasites are there? All of them.
Yep.
And just go through a bunch of different ways, and that they're just animals surviving.
Yeah. The Magic School Bus starts a podcast. Listeners, what would your Magic School Bus episode be? And or pitch us on a Magic School Bus for every episode of the podcast.
Ooh.
Because now that's what I'm thinking. I'm like, well, what else? The Magic School Bus eats ants.
Yes.
Magic School Bus visits the dodos.
Because now what I'm thinking of is, and this for some episodes would be easy, but for some of our like not just a group of animals episodes, what would the bus turn into for like the Bone Wars?
Ooh.
Yeah.
Magic School Bus fights in the Bone Wars. Listeners, let us know what you think. Hop on the social media, or to be part of the broader chatty community, our Discord, which has been hopping. My goodness, our Discord is super active lately. We announced that we reached 1,000 members, a couple months ago? I think, over the summer. We're already at another 100.
Yep.
We are ramping up. Check out the blog post, which will be up on our website, link in the episode description. The blog post will have links to the news articles, as well as links and pictures for diving deeper into the subject. You get some pictures of these cool, weird animals that we've been talking about. Thanks once again to all of the people who requested this topic. This was a whole lot of fun. Thanks to our patrons who support us. Thanks to everybody who listens and comments and discusses in the Discord and shares our podcast with their friends and leaves us reviews. We appreciate all of that feedback and engagement. Extra special thanks to our top tier patrons, Jeff Ellington, Danielle Loves Bugs, and Sarah May. We so appreciate your continued incredible support. Don't forget, this is the penultimate episode of 2025, which means there is one more episode to go. It's going to be a special one. It's exciting. Spoilers, there's going to be another voice on it. Stay tuned. We're excited. Also, at the very end of the year, we will be releasing our end of the year Q&A. Will and I are going to be recording that soon, where we will sit down for hours and hours and answer questions submitted by you, the listeners. And then at the end of January, we will be celebrating the 9-year anniversary of the podcast. We will have a public live stream that anyone is able to join. So come join us for that. We will be announcing the cool stuff that will be upcoming in 2026, which we already have some fun plans. It's exciting times. We release episodes every fortnight here on Common Descent. That is going to continue into the future. So come back and join us at the end of the year for our last episode of the year and the end of the year Q&A and maybe some other special stuff. To be there.
I had the realization that if you are listening to this episode within the first couple of days of it coming out, you may very well likely be listening to it while we are recording the Q&A. So
that's true.
Hopefully that increases your enjoyment to know that we are on an hours-long recording binge of just answering questions.
Well, on that note, I'm going to go get lots of rest. In preparation.
So long, and thanks to all the fish. I thought of that way back.
This is why we pay Will the big bucks.
Bye.
Thanks for listening to the Common Descent podcast. You can follow us on Facebook, Twitter, YouTube, and check our WordPress blog for pictures and links after each episode. Huge thanks to our patrons whose support helps keep this podcast running and who get access to bonus goodies on Patreon. The song you're hearing is called On the Origin of Species by Protodome, which we found at ocremix.org. Thanks again for listening. We hope you'll join us next time.